Awesome, then let's start maybe. So, welcome to today's lecture. This lecture is about nucleic acid design. So this is a part of a two-parter. Next week we will hear about plasmid design, which is of course also nucleic acid design, but in a different way. Nucleic acid. And so when we talk about nucleic acid design, what are we talking about? Of course, the two types of nucleic acids that we have. It's DNA and it's RNA. Of course, in synthetic biology, in bioengineering, we might want to change these. We might want to write our own kind of strings of DNA and strings of RNA. And there are some rules to follow, and this is exactly what this lecture is about. And next lecture is about plasmid design, like how do you put these things together to have your own like bigger piece of DNA in this case. And also today we'll focus on DNA. More or less the computational part for DNA and RNA are very similar. And we will focus here on DNA actually on a very specific topic, which is that of primer design for PCR. We'll come to that in a second, what that means. But just like maybe as a refresher to maybe more computational-minded people, what is DNA? DNA are things like you have these A's, you have T's, you have G's, you have C's. Just writing something here. Right, so this is a piece, a part of DNA. These strings. And chemically, what that means is that you have these nucleotides, and the A comes to the left of the T, and the T comes to the left of the G. And to the left means, in a very specific sense, it's from what's called the 5' end to the 3' end. And it just means that every A, every T, every G is not symmetric. It has a 5' end, it has a 3' end. So, and then you always go like, and we always write it from 5' to 3', and there's a very specific reason for that. It is, or there are multiple reasons, of course, but one of the reasons is that the things are created in that way. Whenever DNA is copied, whenever RNA is copied, it goes from the 5' end to the 3' end. So in the cell, when this is synthesized, it first does the A, then the T, then the G, then the G, then the C, and so on. Always from 5' to 3'. Okay, and you always so know, I guess, that DNA is most often not just a single strand, but a double strand. There's these helixes that form, and maybe you also know what A pairs with. Or the T tends to pair with the A, and the G tends to pair with the C, and of course in the other way as well. And so here we zoomed in on two different DNA molecules. Here the first one we put from 5' to 3'. The thing about the double-stranded DNA is that the other strand goes in the other direction. Here we have a 5' to 3' direction. Okay. That's just how they form. And so this is the DNA word here. And then once we have DNA, we are also already heard in multiple lectures, but in particular in the first one, is that from the DNA template, the RNA gets transcribed. And RNA is almost the same, has the same A, C, and G's, but has a different form of a T. It's a U. It's just chemically a bit different. And what will happen is, if we have like a transcription of this piece of DNA here, it will take a different color. it will start somewhere. So here in the 5' end of the RNA. And we start forming the complement of this T, which will be the A, the complement of this A, which will be not a T, but a U here, and so on. And we know that it goes from 5' to 3', so in this direction is where it goes. So I told you at the beginning that most of the time we write the DNA from 5' to 3', from the 5' to 3' end. And you can see that is making sense here, is that the transcribed RNA is in the same direction as the 5' to 3'. Okay. I might extend this just a little bit. G's here, C, A, U, and U. Because afterwards, of course, once we have the messenger RNA here, we have the translation, and the translation puts the sequence of nucleotides in the RNA into sequences of amino acids in the protein. And it does that by taking triplets. So AUG is the first one, GCC is the next one, AUU is the one after that. And there is just a lookup table. So AUG is always the same amino acid. GCC is always the same amino acid. So you have this 4^3, which is, I guess, 64 different triplets that are possible. And each one of them is mapped either to an amino acid or to a stop codon, which is just meta information, which means please stop here. Okay? And the excuse me, the direction of synthesis of the proteins is here. It starts at the 5' end of the RNA. So kind of it makes sense to excuse me, to write the DNA in 5' to 3' end, because the RNA is the same direction, and then the synthesis of RNA is in the same direction, and then the synthesis of the proteins is also in the same direction. Was this clear enough about nucleic acids, how they form? So the most important structure, of course, not just the linear structure, the primary structure, from left to right. There's more structure to it, which we'll come to in a second. Excuse me. Hey. So what can we do with this? We're talking about nucleic acid design. What is the things that we're designing? Of course, we are designing sequences, but excuse me, for which application? And the application that it's just one particular one, but it's a very important one that I want to talk about today is PCR. PCR is a method to multiply a certain sequence of DNA. And it's a at most it's multiple rounds of a 3-step process. And the 3-step process is, as we have here, denaturation, annealing, and then extension. So what you put into your test tube is just copies of the DNA that you want to multiply. And then you put in what's called the primers. Those are small pieces of DNA, which we will want to design in a second. And then you put in some polymerase, also we come to in a second what that does, and some nucleotides. And we start first just with the DNA, with the double-stranded DNA that we put in. This is what we want to multiply. And at first, it's of course bound to the other strand. It's double-stranded. And what we do first is we denature. What that means is we turn up the heat. The heat is then so big that the two strands will separate just by thermodynamics. And then once we are sure enough that the two strands are far apart, we do annealing. So we turn down the temperature again. And what that does is, first of all, what could happen immediately is that the two strands, again, stick to each other. But before that, we put in enough of the primers here, which are small pieces of DNA that match both strands at different places, but both strands. We put in enough of them so that we hope that the first thing that gets annealed to the two strands is the primer, not the other, not the other strand. And then, so this is, we went high temperature, then lower temperature, then we have two strands, hopefully still separated, with the primers attached to them. These are just pieces of DNA. And then we have the extension step. We turn up the heat in between the two. And then we get this polymerase, which is this machinery that polymerases DNA. So it makes it like a more longer and longer polymer. And this polymerase is found in all cells. All cells have that. E. coli have that. Other organisms have that. And here's a very specific one. It's what's called the Taq polymerase. Taq is a specific organism that lives at high or can live at high temperatures. So its polymerase can live in the high temperatures that we're doing here. So again, here we always want to have a high enough temperature that we're sure enough that the two strands stay separated here. And then it does its thing. So the polymerase sees that here there's a piece of DNA that started, but it has not finished, which is the primer. And it will be helpful enough to repair the other strand here. That's what the polymerase does. And then we're now in a situation where here, starting at the primer in this direction, we have kind of filled up the DNA, the other strand of the DNA for it, in both the template strand and the coding strand. And now we have 4 copies, two exact copies of what we started with. Okay? And so we multiplied by 2 in the ideal case. If we denatured every copy of the DNA, and if the annealing step worked, and if the extension step worked. But in the best case, we multiplied by 2. Okay? And now we can do it again. We cycle again to denaturation, annealing, extension. And each one of these 2 now is multiplied by 2. So now we have 4, then we have 8, and so on. Yeah. So this we can do whenever we have some region of our DNA that we want to multiply. And what we need to do for this are just taking the DNA. This we need anyway. If we want to multiply our DNA, we just need it. We buy the nucleotides, we buy the polymerase. We can just buy them. But we need to have the primers that work with this specific sequence that we have. We need primers, so small pieces of DNA, that can anneal to our DNA here. And there are some rules for the primers that we want. Like we want them we want them long enough and not too long. So if they would be long enough that they have the same length of the strand itself, that would be no good, because then there's no difference between the strands we just separated and the primers. They should also not be too small so that they can bind anywhere. So like if you have just one A as your primer, this can bind anywhere there's a T. So this would be not specific enough. So the sweet spot is of length around 20. But that's just rules of thumb. Okay. Yes. Oh, yeah. That's a very good question. It depends, like, can be a few hundred, maybe a few thousand bases. Yeah. Typically, like, maybe want to like copy a gene or something, and then a few hundred, maybe a few thousand of bases. Yeah. I mean, that is this clear, this procedure? Yeah. So every time the hope is we multiply by 2. Okay. And of course, there are some tools for that. And like more or less in the rest of the lecture, I want to go through a few computational and mathematical tools that we can use to kind of understand how to work with DNA or RNA. In particular, how to work with primers, but it's like a more general kind of set of tools that we can use. And the first that I just want to share with you is what's called BioPython. It's a really handy tool. Can do basic or less basic things. In particular, it can do things that maybe would be too cumbersome to re-implement for us. So here, let's say this is a sequence, encodes like a DNA, piece of DNA molecule, let's say. And we can just have this as a seq here. So this seq is from BioPython. And now we can, of course, compute the length. We can compute the GC content. This is something that's kind of important. Why I didn't mention this, but whenever we have this base pairing, there are two types of pairings between an A and a T, between a G and a C. And it turns out that between G and C, there's a stronger binding than between A and T. So if you would have only G's and C's, you would stick together a lot. If you would have only A's and T's, you might be too wobbly. So it turns out that in nature, there's a sweet spot, apparently, of GC content, somewhere between 40 and 60%, which we also try to do with primers that we just. So here, for example, in this specific example, we have 56.4%. You can also do just very basic but useful things and things that you don't really want to re-implement yourself every time you're handling DNA, like doing the reverse complement. This is a step going from this 5' to 3' DNA to the complement, but in the other direction. Really useful. And can also do like basic things like translating. So you're taking your DNA, or let's say it can also be an RNA string, but here in this case it's DNA, and translating this into protein. So this here now is a sequence of amino acids. Every letter here is a different amino acid. The star is a stop codon. It's the place where translation stops. So if, for example, now you want to have like to write your own, let's say, primer designer, we will come to one that's already implemented. But if you would like to implement something like this, BioPython is a really good way to start. It can also work with with databases, for example. So here, for example, you have access programmatically in Python to like a big database of genes. So here, for example, we're taking this NM000546 gene. It's a gene that's used as defense against tumors in humans. And we can just fetch this, just does a request, gets you back the handle or the record. And then you can see kind of what's the organism about, in which organism does this occur, Homo sapiens, okay? And then you get the whole sequence. So here are the first 50 bases, but of course you can look at the whole thing. Yeah. And then you can just use this. For example, like let's say if you wanted to have like, if you want to multiply this piece of DNA, you can just get its sequence and then do your thing, like find some primers in this sequence. So it's just basic like string manipulation or fetching strings from a server. Oh, sorry, yes. Oh yeah, very good question. Yes, you would always have subsequences, but the challenge of primer design is not to find exactly, is to check where you want to start and how long it should be. So maybe you want kind of this portion of your DNA. You might want to start a bit earlier and finish a bit later, because then maybe you have better thermodynamical properties of your primers. Yeah, but it's definitely always a subsequence because you want them to anneal very well to the template. No. The length, there's a sweet spot between, let's say, 15 and 25 that you also want to aim for, but which exactly you're taking is also a question about the thermodynamical properties of it. So we come to that in a second, but you want to avoid hairpins, that it forms a secondary structure in itself and with the other primer and so on. And then you might want to go for something shorter that doesn't have a hairpin, for example. Even though maybe you would like a longer one, but maybe it is a bad property for you. Right. So the second tool that I wanted to talk to you about is called Newpack. Newpack is like a package, a library that does thermodynamics for you. And more or less the general rule about like thermodynamics of nucleic acids is that there's a lot known about that. It's way easier than proteins. And the reason more or less is that kind of that you almost only need to look at things locally. We'll come to that in a second. But there are, let's say, two big libraries out there. Newpack is one of them. Vienna RNA is another one. And what these things do is like you can put in, we see that in a second, you can put in some piece of DNA or RNA, and then it can tell you what kind of structure it will form. Like, will it form a hairpin that maybe you don't want, or maybe that you do want? Will it stick to another piece of DNA or RNA? And how hard will it stick to the other piece? Just a small word of warning, Newpack is a really good piece of software, but you can't just install it via pip install. You have to go to the website and register, because they have this weird licensing thing going on. For academic use, it's completely free of use, but you have to register there if you want to use it. Okay, let's go through maybe an example here. So here once we install it, we can import Newpack, and then we have the sequence here. Let's say this sequence we want to analyze. And we say, okay, this is a piece of DNA, it's 37 degrees Celsius. And then it can do a few things. And the most basic thing it can do, it can tell you what's called the MFE structure, the minimum free energy structure. So every conformation of your DNA has associated a certain delta in free energy. And the lower this delta is, the more likely it is to form. We'll come to that in a second, how to calculate these. But it assigns to every structure in energy, and the minimum one is the one that's most likely. This is the most basic task, is just MFE. And here we say, oh, please tell us in this sequence what's the most likely structure. So we have GCA and so on, and it ends here with ATGC. And it tells us, okay, here is your sequence. Here's the most likely structure, and let's try to see what this is. So here we have this parenthesis and dots. Dots means it's not paired with anything, and parenthesis means it's paired with, the first parenthesis means it's paired with the last closing parenthesis on the other side. Okay. So this is paired with this is paired with this, and so on. And in the middle, we have 6 bases that are not paired with anything. Okay, let's check whether that makes sense. So here, G and C will be paired. Oh, that makes sense. C and G paired. That makes sense. AT makes sense. TA, GC makes sense. GC. And then here with G and T, and actually these 6 middle ones are not paired in the minimum free structure. And everything can happen, even like off-base pairing, like G can pair with T as well, but of course it's the energy requirement is much higher. It's less likely. Okay. Maybe. Yeah. Just to say 2 words about how these energies are calculated. So you have your sequence here. Let's say your sequence is S1 until SL, the length of the sequence. And the most basic pairing, or let's say, yeah, the most basic pairing would be just to have like the most basic formula for the free energy would be something where we might pair 2, and we only have one additive term for every pairing that we do. So here, for example, there might be one F of S1 and SF, if we decide that these things are paired. It turns out that this is not the best thing to do. Sorry, this plus something else. So like one base to another base thing. It turns out that you need to consider a bit more. In theory, of course, the most complicated would be that everything has like some term with everything else. That you have like n L squared terms because everything asserts some force on the others. This is too complicated, not necessary. It turns out that what is really good is what's called the nearest neighbor model. Which tells you that, maybe let's pick another example here. Pick the first view of here. It tells you that kind of the energy of this conformation is not just one term for every base pairing, but one term for every adjacent pair of base pairings. So here, the stability, that means the free energy delta of this GC-CG pairing is different than a GC, let's say here, TA pairing. That's a bit different. And there's a lookup table, experimentally validated for all these pairs. So kind of the things that you sum up is one term here, one term here, one term here. So that's kind of still a linear number of terms, but it's like one more than you would expect if you just added up all the individual base pairings. And yeah, we'll not go into why that is. In the end, it's just an experimental observation that this is enough. And this has been validated time and time again in experiments. And all this nearest neighbor thing, like the calculation with this nearest neighbor model, is what Nowak does, or Vienna RNA does. Okay. Let's write or read some more code here. So here, for example, we prepared for you some piece of code that can check whether you have some unwanted structures in your primers. So your primers, of course, always come in pairs. One is the forward primer, what's called the forward primer, and the other is the reverse primer, the one that goes in this direction and the other one that goes in that direction. And the things that you want to check, some of the things that you might want to check is if you're putting like twice the same primer, like look at 2 copies of your primer, do they stick together or not? And it might be, right? But like if you have some very symmetric, they might stick together. If you just, yeah, if some symmetry is there. The same with the reverse one. And then does the forward primer stick to the other one? The reverse primer. All 3 of these things would be bad because then they cannot stick, or they are less likely to stick to your DNA template. And here is just a piece of code as we already done it. So here we again looking at the MFE structure, the minimum free energy structure, and we come to why we only look at MFE in a second. But yeah, here we go through all 3, and then we print something if the three structures. And here, for example, we get 3 outputs. So here, these are 2 real forward and reverse primers, and we're checking here. So the first forward primer homodimer, so like forward primer with another forward primer, has some minimum free structure. So minimum free energy structure, as is here. So here the plus separates the 2 copies of our primer. That means that this is paired with this on the on the other copy, and so on. Some of them are not paired at all. The 2 reverse ones also have something heterotime, so forward and reverse also stick together a bit. But here the important information is about these energies, and there are some yes. There are many more. Which can fit. Yes, but it takes the one with the least free energy here. Okay. Not necessarily. Not necessarily, because you have some correction terms that also come into play. Yeah. But the general rule is that. No. Like if you have like a perfect matching between like one strand and the other, you have a very negative free energy. Yes. Because it's very favorable just to bind together. Is that what you meant? Okay, then yes, yeah, sorry. So yeah, and here, for example, here we see -5, -6, and the unit is always here given in kilocalories per mole. And there's some rules of thumb. So here we are looking at the 60 degrees that normally that is a good benchmark to look at, and there is a rule of thumb of around -9 where you want to pay attention. So these are real primers, so kind of they have been vetted, and they are above the -9. So kind of the idea being is that they're good. Like there is some structure, and this is the most likely structure, but even this one is not as sufficiently likely that we care. Not always, but the negative ones are those where you want to pay attention to, because the positive ones will really never happen, except if you turn up the temperature to. Right. Maybe we should also talk about, I think I skipped that, about the form of this delta G. Delta G is the Gibbs free energy, or the delta in Gibbs energy between doing the structure and not doing the structure. And it has 2 components, actually. It's called delta H and delta S. And then it's temperature T. So maybe you heard those terms, maybe not, but it's not extremely important what they are. But just to say that there's one term that's called enthalpy and one term is called entropy. Okay, and they can just be calculated. There are 2 different tables for them. But here, the important thing is that kind of with T growing, you can decrease your whole energy here. So if you have like a high temperature, you can make the different structures almost equally likely, because kind of they will be with very high magnitudes. Okay, so until now we talked about MFE, and what about the other structures? Every structure gets some energy, but then what do we do with that? So here in particular we take again twice the forward primer, and we put it into NUPAC at 60 degrees Celsius, as we did before. But now we're looking at not the MFE structure, but many of the subopt structures, or more of the subopt structures. So there's also the suboptimal structures. It includes the optimal one, but not only. If we just go up again, we see that the optimal forward primer homodimer was this minus 5.53, I should think. And now we're looking at exactly that, but we look at a bit more. So here we take the structure that we already have, but then we see with higher, so non-minimum free energies, we see different structures appearing. And it totally does happen. Like just because this has a bit higher energy doesn't mean that this doesn't happen at all. It just means that it happens at a different frequency. And there is a way to know, based on their energy, how frequently these things occur. So here we do the step from delta G to, let's say, the probability, or let's say the ratio of this structure occurring in equilibrium. So what happens in equilibrium? So equilibrium, of course, we think about longer time scales. Once all the molecules tried all the structures, then and keep changing the structures. So in equilibrium, we do know by thermodynamics that the term S, which is the entropy, will be maximum. This is kind of one of the basic principles of thermodynamics, that entropy goes up, goes up, goes up. And entropy has a formula. Entropy of your system is, let's say, I want to capital N of pi log pi, where the pi's are, let's say, it's the probability of your structure number I occurring in structure. In structure I. So here now we numbered all the different possible structures from I to capital N. And we do know that this is the formula for the entropy, and we know that in equilibrium this will be maximum because entropy goes up, goes up, and goes up. We do know a bit more, which will turn this into a constraint optimization problem. So kind of just to say that this will be maximum. We maximize this, subject to. We do know, of course, that these being probabilities, they will sum up to 1. Everything has, your system has to be in some structure, in some configuration. So the sum of the pi's will be 1. we also know, and now this goes back to the energies, that the energy doesn't change. So Pi times EI, where this is the energy delta of being in one of these structures, in structure I. This has to be a constant, let's say. It's the average energy in your system. This is just a constraint optimization problem that we can kind of know how to work with. Here we, of course, we will use Lagrangian multipliers. So we'll put the Lagrangian to be the optimization, the function to be optimized here, which is some Pi log Pi. And then we have the lambda of the first constraint being 0, Pi minus 1, let's say mu, and this is the second constraint, Pi EI minus E. Yeah. Then we know that kind of all the partial derivatives have to be 0 at the optimum, and refers to the PIs, and this will be enough. So this will be 0. So here let's do this with respect to Pi. So the first one here, so the other PJs don't matter, so only Pi does matter here. And this is just a product of two things. The first one here, right, Pi differentiated with respect to Pi is just 1, and then we have log Pi remaining. And then we have like the Pi times 1 over Pi, which is the derivative of log Pi, which is just 1. And here we have only lambda times Pi here, and then we only have the Pi times EI here, which is mu. Oh, excuse me. Of course, will disappear. And here we only have the EI. And now we can translate this back. And now we can know what the correct expression for Pi is as a function of EI. So if we know the energy, what is the probability? Then once this is the probability, it's also the ratio of this structure occurring in equilibrium. Here, can we do? We can, sorry, I think I forgot a minus here. And minus here. Yeah, because of this minus. Okay, so we take the log P to the other side. Pi equal to lambda minus 1 of mu EI. And we just exponentiate both sides. So this is E lambda minus 1. This is E mu times mu EI. And this tells us something about the form here. So here, this we will just call 1 over z for a reason that will become apparent in a second. And then we can have that this is e power some constant mu times EI over z. Once we know this, we immediately know what z has to be. Z, of course, has to be the sum of all of them because they have to sum up to 1. And Z is also called the partition function, and Nowak can also work that out for you, what the partition function is, what the Z is. It can work out for you what EI is, and it can thus also work out for you what the probabilities are, and we come to that in a second. Which only leaves the question about the constant mu here. And it turns out that mu, and this is not extremely easy to show, but it can be done, that mu has to be minus 1 over Kb times T. T is the temperature, Kb being the Boltzmann constant. And then we have our formula. Then we can go, so these are just, Kb is just a constant, and T is just the temperature that we have to know. And then we can go from the energy to the probability, and thus the ratio of the structure occurring. And we also see that the energy is in the exponent of this. So it stands to reason to think that a small difference in the energy might have a large impact on difference or ratio of the probabilities. That's indeed what we see. And this kind of gives some credibility to the approach of only looking at the MFE structure all the time. It's still possible that there is another structure with maybe the same or extremely close energy right next to it. But as soon as there's some separation between the two, it's reasonable enough to assume that MFE is a good starting point. And I made this calculation with Nowak for you, just based on this structure that we just saw. And Nowak, of course, can't do this calculation for you. So here we look at the structure probability for each one of the structures that we saw right above in this subopt compact collection. And this is the MFE, and those are the rest. So the first one has like 1.6%, and the next one has then 0.5%. So there is a clear separation between these. Yeah. I mean, so it's so one thing, just a few observations. So I can't give you the answer. But one observation is that you never see these numbers being really, really high. Like 10%, 20% is already very high. So yeah, maybe if you see 20% here, you might want to reconsider. In the 1% range, it might be okay. Yeah. But you almost never see it being close to 100%. That's because you almost never see them being close to 100%. This is too much to expect. But a really bad, a bad primer might be here, like with the structure in the 20s or 10%. Yeah. Yeah, of course. Oh, this is the so whenever you have like a constraint optimization problem like this, you can find an optimum at these points where all the partial derivatives are 0. So with respect to the original coordinates here, pi, but also we use this implicitly but not explicitly, like with respect to lambda and with respect to mu as well. And yeah, it's a very short proof. Normally, this is done in real analysis 1. Just turns out. Oh, the Lagrangian multipliers. There are 3 variables like this. Afterwards, you will know the values. So for example, for lambda, we found out the value by using the fact that lambda L over dl over d lambda has to be 0. By using this. And by using the other and some more arguments, you can also fix down. Okay. So this was some thermodynamics. Then we can do a bit more with. So once we've taken care of the thermodynamics and we're happy with it, meaning that there's no secondary structure, no harsh binding between forward primers and reverse primers and so on, there are a few other things to check. And one of the things to check is what's called blasting. Blasting is you take a piece of DNA and you compare it to all the DNA in some organism. And you can do this locally, might take some time. There are also web servers that do it for you. And this is also a good idea to check what's called specificity, so that you're only amplifying your gene that you're interested in, not too many others. And here you see, this is built into BioPython again. How you do that. Just a QBlast function here. And we're blasting our fault primer that we had before. And yeah, this takes a minute. And this is kind of accurate. It takes about a minute. And then we come back with some of the results. And here we're blasting against Homo sapiens, which is a reasonable target because this is a forward and back reverse primers for a gene in Homo sapiens. Then we come back to us and tell us what similarities it found. And there's a specific value that it will return, which is called the E value. And if it's lower, then the match is more significant than if it's higher. But we'll see that in a second, I hope. Sorry. Oh, no. So this is not the matching. The matching is between this and what comes after the plus. Yes. No, not necessarily. So for any binding that you can write down, eventually it will occur. If you wait for millennia and look at your two different molecules, it will occur at some point. And anything. So even, I don't know, like this binding to this will happen at some point, and nothing else binding will happen at some point. It's just a question of like, how fast does it do that and how often does it do that? They might, right? And so this is why this has a smaller free energy. It's more likely to, but still it can happen that it doesn't. If you take a snapshot, it can happen that it currently doesn't. So this is the whole thing, right? So everything happens. It's just a matter of probabilities. They might. Yes. And for example, like if you have a perfectly matching DNA, it will bind the whole way most of the time, but it might also breathe sometimes. Yeah. No, it's a game of probabilities. Yeah. Sorry. Now we're back with our blasts and we get some exact matches, which you go through them. For us, it's not a big problem because they all pertain to the gene that we want to target. But in general, you might have some surprising finds here. So here we see really small E value and identity 24 of 24, which means that our original thing that we searched for was 24 nucleotides long, and all of them occur somewhere, which is to be expected here because this is the gene that we were targeting. Okay, but it's good. It's a good thing to blast your DNA against the organism where you want to insert them into, whose DNA you want to work with, because there might be some off-target things that you didn't think about. Okay. 10. So the first one is the target, and the others are also associated to that target. So all of them here in this specific set of hits, it's fine. Those are perfect matches, but the matches that we wanted to have, or that we were fine with having. Yeah, and towards the end of just to wrap this thing up, is that of course people have like built explicit primer design libraries as well. The most famous one is Primer3. As a web interface, also exists as a Python library. It can do some things for you. It can do everything that Nucleic can do for you, but it can do quite a bit. And here, for example, there's a function like a sequence and where we want to target the start and the target to end. And here you can set some of the parameters. For example, like how big should your primers be? And again, there's like this sweet spot somewhere between 18 and 25 nucleotides long for your primers. You can also specify like an optimum size, whatever. Like an optimum melting temperature, GC content, and so on. Then we just go and find these things for you. So here, of course, there is some target sequence that we might want to amplify. And just ask it to amplify in this target sequence the subsequence between 200 and 400. And this comes back really quickly and tells us, oh, I found a really good set of primers. And they have a really similar melting temperature, have very good GC content, and the length is 20, which is our optimum. And yeah, and then you might want to look at this in more detail, maybe blast it, other things. Yes. Yeah, it might bind too hard, so you might have a lot of secondary structures in this case. So because G and C bind harder together, that also means that it's more likely to form these things. It's 100%. Yeah, and then again, like the sweet spot seems to be somewhere between 40 and 60%. That's where you want to end up. Right, and this is almost everything that I want to tell you. Just like you can design your primer with Primer3, you can also inspect a primer that is given to you. So here, for example, is a small thing that checks a primer, checks like the hairpin formation, like delta G, hairpin formation, homodimer, delta G. So here, for example, for this specific one, it tells us that there might be a problem because we have a really small energy, so really likely to happen homodimer going on. If we take this, it binds to itself, really sticky. Also, the hairpin is a bit too harsh here. Of course, this primer is not a real primer. This is just a random DNA sequence. If we take the primer, the fourth primer that we worked with until now, which was a real primer, we get much better numbers here. So there's no hairpin with negative energy even, and homodimer is around -3, which is good. It's far away from the -9 that normally we take as a warning sign. Yeah, and that's it. Just to wrap up. So we looked at a few computational and mathematical tools for design of nucleic acids, in particular primer design. But the other nucleic acids that we want to look at and that we want to design, in particular plasmids that we look at next week. So in terms of computational tools, 3 things. BioPython, really useful, it does basic things, but it's always awesome to have because it's also standardized and just does things for you. Nucleic for thermodynamics, really useful. Implements the nearest neighbor model. Can do amazing things for you. Different structures, not only the MFE structure, but also suboptimal ones, and associate the probabilities to it, and so on. And the blasting is also included in BioPython, very useful, and Primer 3. You will explore that in the prom session, but it's a really useful tool just to give you some primer. Then, of course, you might want to analyze it a bit more and see whether there might be any problems blasting and other things, but it's a really, really good starting point. Do you guys have any questions, comments? Is there anything online or no? Okay, I just heard something. Yeah, okay. Okay. And thanks. And prom session is about primer design. Design a primer. You will find some problems in primers that we give you. Yeah, and looking forward to the prom session.